Welcome back. The coronavirus is, like any other viruses, not much more than a shell around genetic material and a few proteins. To replicate, it needs a host in the form of living cell. Once infected, this cell does what the virus commands it to do: copy information, assemble it, release it. But this does not go unnoticed. Within a few minutes, the body's immune defense system intervenes with its innate response. Granulocytes, scavenger cells, and killer cells from the body and lymphatic system stream in to fight the virus. They are supported by numerous plasma proteins that either acts as messenger or helps to destroy the virus. For many viruses and bacteria, this initial activity of the immune system is already sufficient to fight an intruder. It often happens very quickly and efficiently. We often notice only small signs that the system is working. We have a cold and a fever. Interferons are subgroup of signaling proteins that are normally secreted by infected cells. SARS-CoV-1, which is responsible for the SARS epidemic in 2003, appears to have suppressed the production of one of these interferons and thus at least delayed the attraction of immune cells. At a certain point, however, the host response is so strong that its effect can be counterproductive. For example, numerous immune cells can enter our lungs and cause the membrane through which oxygen normally passes from the air into blood to thicken. The exchange of gases is restricted, and in the worst case, ventilation may be necessary. Sometimes, the reaction can overshoot and be directed against healthy cells as well. This could also be the case with the novel coronavirus. So, drugs are also being tested that suppress an excessive immune reaction and that are already known from the treatment of autoimmune diseases. The balance between protective and overly aggressive immune process in dealing with the coronavirus is currently a big mystery. After a time delay, the acquired immune system finally sets itself in motion. It is different from every person and depends on what we have experienced and which pathogens we have come into contact. While T cells help destroy infected cells, B cells form antibodies that can keep the virus in check. In the case of coronavirus, these are neutralizing antibodies that bind to the spike protein of the virus. This is the site of attack of the virus with which it enters the host, that is our human cell. The good news is that it is very likely there is an immunity. This is suggested by the proximity to other viruses, epidemiological data, and animal experiments. Researchers infected 4 rhesus monkeys, a species very close to human, with SARS-CoV-2. The monkey showed symptoms of COVID-19, the disease caused by the coronavirus, developed neutralizing antibodies, and recovered after a few days. When the recovered animals were reinfected with the virus, they no longer developed any symptoms. They were immune. It is not yet known how long the immunity will last. It depends on whether a patient has successfully developed neutralizing antibodies. Akim Horowitz estimates that the immunity should last at least 1 year. Within this year, every new contact with the virus acts as kind of booster vaccination, which in turn might prolong the immunity. Researchers are already collecting plasma from people who have successfully survived an infection with SARS-CoV-2 and are using it to treat a limited number of patients suffering from COVID-19. The studies carried out to date have shown positive results, but they have usually been carried out on only a few people. PCR tests are currently used to find out whether a person is infected with the coronavirus. With the help of PCR, it is not possible to tell whether or not there is a reproducible viral RNA. It is just a proof of whether the virus is still present, dead, or alive. According to all we know, it is not possible with the same pathogen, says Akim Horowitz. It is possible to become infected with the other coronavirus or viruses from the SARS or MERS group if their spike protein look different. As far as the current epidemic is concerned, it can be assumed that people who have been through COVID-19 will not become ill from it for the time being and will not transmit the virus any further, he says. A study carried out on the first coronavirus patients in Germany showed that no viruses that are capable of replication can be found from the day 8 after the onset of symptoms, even though PCR can still detect up to 1 lakh gene copies per sample. This could change the current quarantine recommendations in future. While some people get off with a mild cold, others are put in ventilators or even die of SARS-CoV-2. Especially people with pre-existing conditions and older people seem to worst affected by the virus. Why? This is the hottest question at the moment. It will still take a very long time to understand the mechanistic, biological basis for why some people are so much more severely affected than others, as said by virologist Angela Rasmussen. The virus is important, but the host response is at least as important, if not more important, her colleague Stanley Perman told the magazine. Thanks for watching. Make sure to give us a like and also don't forget to subscribe. And not just subscribe, ring the bell so that you don't miss any video from us.